Firstly, over the last 10 years, the most discussed topic about healthy food and individual training approach system. The workout, treadmill, and elliptical are popular aerobic loads which are able to accelerate metabolic process and straighten our cardiovascular system. However, not everyone can lose weight and achieve the desired result during treadmill. He noted that personal features of nervous, endocrine system, metabolism, and kind of lifestyle influence on the result of training, but nobody ever realized the importance of sex hormones effect. Which one? On this slide, you can see that sex hormones are able to affect on water balance, changing oxidation in muscles, and regulate fat metabolism. Notice, estrogen and testosterone effects are opposite to each other. Bioimpedance measurements is the best way to record all changes in biological tissues during aerobic loads. This method is based on ability of biological tissues to conduct an electric current. The resistance of tissue to electric current depends on the content of liquid in them. The higher hydration of the tissue, the better conduct it could be. That's why we poorly hydrated adipose tissue is an insulator. Bioimpedance analysis of body content allows to control the state of lipid, protein, and water metabolism of the body as indicators of effectiveness of aerobic loads. So that's why the aim of our study, of our research, was to determine sex differences of bioimpedance measurements of visceral body in response to prolonged aerobic loads. For the study, animals were divided into 4 groups, 2 control groups, and 2 treadmill train for the next work rate. On this slide, you can see the method of our research we used. Treadmill trained rats had measurements of work rate on the first 7 and 14 days. The animals ran at the speed 12 meters per minute for the first 3 minutes. The acceleration of treadmill movement by 0.6 meters per minute squared was activated after 3 minutes of running. So it was found that the female rats lose weight at 14.9% in comparative to 7 days, whereas males did not significantly throughout training period. Female rats had significantly increased heart rate by 5.4%, which was probably due to the activation of adaptation system, sympathoadrenal actually. The blood pressure, no significant changes. It was found that the work rate of males was 16.3 minutes on the first day. Aerobic loads on the 7th day resulted in an increase 1 to 18.4%. And it should be noted that despite the positive result of training in the male group, the work rate was characterized by a heterogeneous result among rats. One set of males have a low work rate due to aggression, refuse to run, and sit on the electrodes. The other had some minimal positive changes, and the last third showed an effectiveness result. The work rate of females at the beginning of training did not differ significantly from the result of males. On the 7th day, they showed an eventually effective result in a group which exceeded the initial values by 44.2%. On the 14 days of learning, the male rats showed high stamina like a female rat. The bioimpedance results showed male has a higher percentage of total body water and intracellular fluid compared to the female, while female had a higher count extracellular fluid. Males had a higher percentage of free fat, whereas females had a total fat percentage. After 14 days of training, the pattern of water and fat distribution changed again to the opposite. In males, the percentage of total fluid decreased by 9.8% and increased extracellular by 15.3%, while female had the intracellular fluid increased by 14.2% and extracellular decreased by 5.4%. Prolonged aerobic load has shown that sex hormones significantly alter the nature of fat distribution in the body. In this case, males had an increase of 49.2% of total fat, but a decrease by 20.5%. In females, the changes were opposite. Total fat decreased by 22% and free fat mass increased by 11.44%. So as a result, I would like to say that the influence of aerobic loads on the male and female body is significantly different. There is a change in the water balance in the male rats, while in the females, we see a significant effect on a fat metabolism. Also, with the help of training, it's very important to take into account the initial states of the water balance and fat metabolism for correction of food and water intake during the training. Thank you for your
Thank
attention.
you for your speech. Thanks a lot. Please, members of jury, you can ask the questions. Are there any questions to the contestant?
Yes. Anastasia, thank you for your report. I'm little bit shocked by your result. How is it possible? But interesting. First of all, how do you think? Why this difference? According to your result, after physical cardio loaded, male not lost kilogram, not lost fat, but increased fat mass. How is it possible, and which factors it can be depends on?
Okay. As you remember, I told about some troubles with trainings when females just sat on the electrodes and they refuse to run. And that's why I think that it's have some influence on their level of catecholamines and their level of testosterone, and that's why they was in stress. The females isn't stressed up as the males want, and they a little smarter than males. That's why they we're gonna talking about the better adaptive better adaptation through the females, but the worst one for males. That's why they have unfortunately increase of fat mass.
Okay, interesting. And I have one question more. Maybe you found somewhere. What about some norms for extracellular and intracellular content of water? We have some norms for this, or no?
And as it, you didn't hear me. You
Yeah. Okay.
Look, I showed the result. If you want, I try again to show you. Yes.
Okay.
Wait, please, if you want. Where is it? Do you see this one? The results. Hello.
Yeah, we can see.
Don't worry, continue, please.
Okay. Look, these amounts we have, this is the average for male thread, and that was described in many publications. You can see it in NCBI. I have a lot of literature in my work. At the end of the list of literature, I do the body water content in males. That's between 1 per 1, intracellular to extracellular, and the total and the more count of extracellular fluid in females. I could describe like influence of estrogen, and that's really important because estrogen are able to influence not only lipolysis on lipolysis and fat metabolism, but on a water count exactly. And I read about it a lot, and a lot of publications show that this really depends on the level of estrogen. In publication of Italian maybe, they described the difference in a human in humans which trained different aerobic loads. One used to just work out, another elliptical, and other one use nothing. I mean, they're not trained at all, and they have the same at the beginning level of extracellular fluid, and it's really always higher than intracellular, and they trying to find out the level of cholesterol and lactate and estrogens, and they find that when the level of estrogen is higher, the content of fluid extracellular become more higher.
Okay, thank you so much. I wish in your next investigation you can do the same experiment maybe with people already, and maybe you can measure more indexes inside blood, for example, or level of hormones for more express your results. Thank you.
You know, I have a lyophilizate, but unfortunately quarantine, and I'm seated on another city, so I just lost my time, and I'm
And continue.
Okay, thanks a lot. And we have more questions from the chat. The question from Alexander Birchanov is more or less the similar about the differences between male and females, and your explanation was okay. And the next question is, whose idea was this study?
This idea was mine because I have some troubles with loss weight, and I didn't really understand what I can lose because I trained 3 times a week, and I have some diet, but I realized that this week I trained, I've not lost any kilograms, and I just surprised, but I feel that I was exhausted and I'm working hard, so that's why this put me to this question and to describe it, to research this one. I think this question upon to every one of us.
Okay, thank you for the answer. And I have one more question. If you, as you should have known about from the pathophysiology course, that total volume of water in human body is about 60%. Actually, it's 72.3 of ideal body mass, but let's say 60%. 2/3 of this is intracellular water, and 1/3 is extracellular, which is not the same to your data in your work. So how can you use your results? How can you implicate your results towards the humans? Because we have different water proportion. It's not the same to rats, as you can show us. Have shown. So what can you tell us?
Yes, I understand that we have different count of water balance, but the distribution of water balance and distribution of fat mass is the same. That's why I was fixed up not on account of water, but the way and which parts and which substances are able to influence on distribution. This was the main.
Okay, but as I would see, as I would saw, it was like 50 and 50, right? Extracellular and intracellular in male, but in male human males it should be 2/3, 75%, and 20-25% extracellular. It's not the same.
But
The proportion, the ratio is not the same.
Yes, it's not the same, but I could ask myself the question: is the bioimpedance of human body is a real significant? Did you see
So-
this weight loss, not weight loss, weight count? How does it look? And we don't take needles into our cutters, how I made to the rest. And I have the full connection, the current, and better conduct the current than if we try to make this one on the human body with our bioimpedance analyzer. Maybe this may have some influence on it. Really?
Okay, thanks a lot, but just in case, my idea was if you want to have some implication to humans, maybe we should study humans, because the rat model cannot represent all the peculiarities of human body. Thanks a lot. Are there any more questions?
Yes, I have one question. How does your result depends of age, and what age of animals which you used in experiment? And if yes, what you can say about when you take in experiment older animals? How do you think about this?
I understand. We use 72 weeks.
Approximately, or close to what is the age of?
Close to what?
What is the age of animals which you used? Not exactly, but for how many months you used?
Our rats is above 7-8 months. 7-8 months. It's adult age, not the old rats. I know that if we're talking about more younger, the count of fat mass it's so less, and there are a lot of water mass in young, and so young mice and rats. But we always use rats young, not elder, because they have maybe some pathologies like gross pathology or something else, and it could trick our results. That's why I use younger.
So, such as I understand, all of your result you combine with the influence of hormones, these were sex hormones like estrogen and testosterone, yes? And with age, this amount of hormones can change, of course, and this can influence. Maybe you continue your research and try to study with age too. Thank you.
Thank you for the questions.
Okay, thank you for a great discuss.